Hi all, welcome back to our Midbrains channel. So this video is about major histocompatibility class 1 mechanism or simply MHC class 1 mechanism. So we are going to discuss how an MHC acts inside a cell when an antigen enters, how this cell then expresses to a Cd8 T-Cell. Important organelles related to or needed for this MHC class 1 mechanism or activation are endoplasmic reticulum, proteasome, ribosome, Golgi apparatus and so on. So now let's discuss its mechanism. When a pathogen, let's take a bacteria as an example, enter the cell, the pathogen will make its products like proteins inside the cell. Then these proteins, exactly saying the pathogenic endogenous proteins, enter the proteasome. The function of proteasome is to degrade the cytosolic proteins into protein fragments or peptides. So these pathogen proteins are then broken down into small peptides. These small peptides can now enter inside the endoplasmic reticulum through the TAP transporter. So now let's hold it there. On the other side of endoplasmic reticulum, we have the ribosome. This ribosome makes protein from the RNA by the process known as translation. So these ribosomes now make MHC class 1 protein. But initially, this MHC class 1 proteins will be incomplete with only alpha 1, alpha 2, alpha 3 domains. This incomplete protein is bound to another protein called calnexin. This calnexin acts as a chaperone, that is, it assembles or make these proteins reach their destination. There is another protein called microglobulin beta 2M which attach to the incomplete MHC class 1 protein. So now we have a complete MHC class 1 proteins made up of alpha 1, alpha 2, alpha 3 and beta 2 domain. At the same time, there are some other proteins attaching to this MHC class 1, specifically calreticulin and ERB57. They function as a covering for the binding site or groove of this MHC class 1 protein. So this will prevent other peptides or antigens to bind or interact with the binding site of MHC class 1. These proteins too act as a chaperone like the calnexin and bring the MHC class 1 protein near to the TAP transporter. At here, this MHC class 1 will interact with another protein called tapasin. When MHC class 1 interacting with tapasin, the calreticulin and ERB57 automatically dissociates. So now the pathogenic peptides in the endoplasmic reticulum can easily bind at the binding site of MHC class 1. After this binding, the MHC class 1 together with the antigenic peptide leave the endoplasmic reticulum within an endosome, and it will go through the Golgi apparatus by a normal exocytosis process, and it will then from the Golgi apparatus in an endosome, the MHC class 1 together with the antigenic peptide will be presented on its cell surface. So this cell which is affected by the pathogen will express its MHC class 1 with a small fragment of the bacteria on its cell surface. They then wait for the interaction with Cd8 T-Cell, a cytotoxic T-Cell. The cytotoxic T-Cell or Cd8 T-Cell have few receptors like Cd8 receptor which interact with MHC class 1 molecule. The infected cell presents the antigenic peptide to the Cd8 T-Cell. Then the Cd8 receptor checks if it's an MHC class 1 being expressed. The Cd8 T-cell has another receptor called TCR or T-cell receptor. It checks the antigen because the T-cell receptor only fits to a specific antigen. Now, if both are okay, if the Cd8 receptor recognizes it as an MHC class 1 and the TCR fits to this specific antigen presented, the cytotoxic T cell then produces chemicals, cytokines, and they will kill this cell. Hope you liked the video. For more medical related videos, get your free pass by hitting the subscribe button. And thanks for watching this video.